Welcome. So, in next of our few classes, we will discuss the corresponding enzymes of several metal ions such as molybdenum, tungsten, vanadium and chromium. These are different compared to what we have discussed so far, and today we will just focus our attention on this particular one, that means the molybdenum, which we all know that it is the only second row transition metal. Second row transition metal, that is essential for several living organisms. So, what this particular one can do, which we will see particularly from the molybdenum coordination chemistry, if we know little bit of that, because this particular metal ion is most abundant and sometimes is very much found in sea water. And it has give corresponding complexes in two most stable oxidation states, one is +4 and another is +6. So, well known metal complex of molybdenum, we all know that MoO2 species having, if you have acetyl acetone, ACAC we call acetyl acetonate ligand, which is nothing but your this one. So, when it is bound to that, that means if you have the molybdenum compound, so these two ligands, this is basically your O donor ligand, so which will bound this OO and that OO, and you have the corresponding dioxo compound. So, this molybdenum dioxo compound, which is immediately tells us that it can be stabilized in +6 oxidation state. So, if we can go for a 2 electron transfer from that compound, we will get or we will stabilize this corresponding molybdenum compound or molybdenum present in the biological system, because this one, if it is abundant in sea water, but are rare in the earth crust. In the earth crust. So, if we can go for settling between these two oxidation states of +4 and +6, and obviously, if we can remove one of the oxo part, so that can be stabilized. If it can go for binding to water, you can say that it can be stabilized to +4 oxidation state. So, the intermediate one, which is +5, is sometimes between the catalytic cycle, is also accessible. So, we can have these 3 oxidation states. And what we are going to look is that one such interesting reaction what we have studied for iron chemistry, that hydroxylation. But this sort of hydroxylation is different compared to other hydroxylating reactions, because water we will use here as the source of oxygen if we at all go for any kind of such hydroxylation reaction, because in the different monooxygenases what we have seen in case of iron chemistry, that we had monooxygenases and in those monooxygenases, instead of this water molecule we have used O2 as our source for oxygen. So, this O2 is basically hydroxylating our species, such as if we had the substrate what we have seen earlier, then that substrate can be hydroxylated, SH can be hydroxylated to SOH. So, any kind of that substrate utilizing water forming H2 over there. So, water, then 2 electron and 2 proton. So, side by side while we talk about all these molecules, we will compare what we have seen in case of the iron chemistry and what we basically expect when we change the metal center from iron to molybdenums, because this particular one is different, because this oxygen from O2 is incorporated within the substrate or into the product. So, these monooxygenases what we have seen in case of dopamine beta monohydroxylases, then cytochrome P450. So, all we have studied earlier, but we should be able to compare one after another. This is dopamine hydroxylases and then methane monooxygenases, all are utilizing oxygen from the air or any other oxygen source, but in this particular case you will be utilizing water molecule as a source of oxygen. So, that means water can be able to give you, that means if we can go for the attachment of the water to the molybdenum center, or at some point it is possible to attach the OH group to the molybdenum center, or ultimately the oxido group, that means the oxido ligand to the molybdenum center. So, these are all interchangeable. Once you have the water molecule, you expect that at some point you can generate the molybdenum oxo compound or all species on the molybdenum center or the corresponding hydroxido species. So, this particular classes of molecules, we will call them as molybdenum hydroxylases. So, these molybdenum hydroxylases which are therefore different from our iron monooxygenases, and they are basically responsible for the corresponding introduction of OH function on any carbon. And one more interesting thing is that they are basically operating on different eukaryotes and prokaryotes. Eukaryotes and prokaryotes. Because in the case of iron corresponding transfer mediated by iron, we have the environment or we have the atmosphere which is dominated by O2, that means you have aerobic environment, but in case of molybdenum, when oxygen supply is less and we have the water corresponding water molecule, and that water molecule is responsible for the corresponding hydroxylation reactions. So, one such example is that we call considerably that molybdenum oxotransferase. So, molybdenum oxotransferase is, the name immediately will tell you that if you have the molybdenum and the corresponding molybdenum oxo compound, and that oxo can be generated from water molecule or the hydroxide of function. So, you have the corresponding molybdenum oxo species, and that molybdenum oxo species can go and transfer to any other substrate. So, therefore, this is oxotransferases is basically a superfamily of molybdenum oxotransferases. All contain molybdenum and promote oxygen atom transfer reactions. But interestingly, at that point you should know that this oxygen transfer basically is taking from the water molecule. So, there are a large number of these molecules are present, and we will see 2 or 3 examples for that. One of them is dimethyl sulfoxide reductase, then xanthine oxidase, nitrite reductase and sulfite oxidase. So, all these things what you see, that basically what we are talking about nothing but only the transfer of oxygen. So, if we are able to transfer this oxygen, what we are unable to do without the help of these enzymes, that means from the water molecule itself. So, if you have this dimethyl sulfide, so how you can go for the corresponding transfer of dimethyl sulfide to dimethyl sulfone and then dimethyl sulfoxides. So, how you transfer that oxygen on this sulfur, because we all know they are very easy to get, particularly when you have the thioether molecules, and even the simple hydrogen peroxide can transfer one such oxygen to the sulfur center. Then xanthine oxidase is also basically the corresponding oxotransfer reaction on the xanthine molecule. Then nitrite reductase, basically it is the interconversion between nitrate and nitrite. So, always we are talking something related to how you transfer the oxygen atom from the water molecule through molybdenum to the nitrogen center, to the sulfur center, to the carbon center, or the corresponding sulfur center in the sulfite anion. That means, the sulfite to sulfate interconversion is also possible with the help of these molybdenum oxotransferases. So, we consider them as cofactor, because this cofactor is required for the activity of the enzymes such as sulfite oxidase. Xanthine, we can call it oxidase or oxidoreductase, because the reaction is reversible one. And one other example can also be your aldehyde oxidase. So, aldehyde molecules can also be oxidized, because you can transfer that oxygen to your carbon center of aldehyde. So, it is a corresponding, we call it as a coordination complex basically, because you have the definite ligand system which is bound to the metal center, and we get it as a molybdopterin molecule, and that molybdopterin molecule which does not contain any molybdenum. So, that is why we call it as a molybdenum cofactor and an oxide of molybdenum. So, in one point you have the oxide of molybdenum and another cofactor is required. So, what is that? So, this is your molybdenum factor. So, this particular part is derived from some other thing, that how it derived, we will also see that this is without molybdenum, but since it can catch up molybdenum very nicely, we call it as a molybdopterins. When reacted with molybdenum or tungsten, because little bit we will see about the tungsten also, because you know that the chromium, molybdenum and tungsten, they have the similarity. So, if molybdenum can give rise to such species or such compounds, we expect that tungsten can also give all these molecules. So, form a molybdate or tungstate. So, basically not any bare molybdenum, because molybdenum always has some affinity for the corresponding formation of molybdyl species. It is very difficult to get any molybdenum compound without your oxo center. So, always they go for the corresponding species as the molybdate and the tungstates. Are class of cofactors found in molybdenum and all tungsten enzymes. So, This is basically we call it as a molybdoterdin, and it is basically the one such cofactor. And when we see the entire molecule, we always try to find that what are the groups basically present which can go and bind to the molybdenum center. So, these are the two sulfurs. So, you see that this particular part, so this particular part is basically suitable because this particular part, so S, this S and the other S. So, sometimes it is very difficult to identify what are these R groups, whether these R groups are hydrogens or any such alkyl function or alkyl group. But the positioning of these two sulfur atoms immediately tells us that this can behave as a very good bidentate ligand, like that oxomolybdenum acetyl acetonate compound. So, if you need always a bidentate ligand system, whether it is based on oxygen oxygen bidentate ligand or sulfur sulfur bidentate ligand. So, this molybdoterdin molecule is basically functioning, is a whole big molecule, but only this particular part will go and attach to the molybdenum center, and it is behaving like a bidentate ligand. So, this bidentate ligand then go for the functioning of the corresponding enzyme function, whether it is a xanthine oxidase or it is a sulfite oxidase type of reactivity. So, it is consisting of a pyranoterdin, a complex heterocyclic featuring a pyran. This is the pyran part. Is the pyran part is fused to a pterin ring. So, basically in the biological system they are biosynthesized. So, in biological system we generally in situ we are generating this species, and this pyran ring is important because on this pyran ring we have the disulfur function which can be utilized for molybdenum coordination. So, this pyran ring therefore features two thioethers which serve as ligands in molybdo and tungsto enzymes. So, why we call it as a molybdoterdin part? So, this pterin part is the you have this other part. So, other part is the pterin ring, and which is sometimes we call it as a pyranoterdin ring also. So, the pyrano ring where you have two sulfur groups attached to it, and those two sulfur groups are basically utilized for molybdenum coordination. So, what basically we get from there is a typical system. So, molybdoterdins are nothing but a useful ligand system. So, is a dithiol ligand. So, like that of your ethane dithiol, what we can have. So, is a dithiol ligand and which can be generated in situ. So, in the biological system, so that can be biosynthesized, and interestingly all other groups, basically your nitrogen and oxygen and all other parts of these groups, so they are having also potential donor groups or donor functions, but those groups are not utilized for molybdenum coordination. Only these two sulfur groups have higher affinity for molybdenum binding, and this particular part is responsible for molybdenum coordination. So, that we get from this particular one ring fused to the other, and the entire molecule is responsible for coordination to your molybdenum system. So, this particular when it bonds to the molybdenum, you see now you have this. So, this is basically giving a bidentate coordination from these through these two sulfur atoms, and these two sulfur groups, if they are bound to hydrogen, they can remove very nicely depending upon the pH of the medium. So, they can be converted to the thiolate function, and those two thiolate groups are bound to your molybdenum center, and this molybdenum dioxo center, these molybdenum dioxo groups are very useful, and in this particular case you see that molybdenum can move from a coordination number of 6 to a coordination number of 5. So, what we all know that any catalytic function like that based on palladium, that palladium can have a coordination number of 4 and it can go up to a coordination number of 6 also. So, here also molybdenum, when it is not in the highest possible oxidation state, but in this particular case you have these two thiol functions in the S minus and S minus and molybdenum dioxo form, and this can be some other loosely bound group from the enzyme system. So, our human body contains very less amount of molybdenum, but their catalytic functions and all their reactivities are slowly we are going to know all these things. So, per kilogram body weight we have only 0.07 milligram of molybdenum, and mostly it is available in liver and kidneys, and it is in the lower concentration in the vertebra. Molybdenum is also present in human tooth enamel and may help prevent its decay also, because in the solid form. So, in some molybdate form it is sometimes little bit it is present within that of your tooth enamel. So, when we have this molybdenum and when we identify that this particular molybdenum is at least bound to a diethyl system. So, is a bidentate coordination bidentate binding from the organic ligand part is available. So, basically we will talk 3 such groups of molecules or 3 families which are catalytically active, and these 3 families, the first one is basically that of your xanthine oxidase, which we consider as XO, and here we have the molybdenum center, and is not only sometime bound to your oxo form, but is also from the sulfide function also it can be present, and OH is also present, and you have that from the molybdotyrine part, the sulfur part, and this can settle between hydroxide function and water function. So, once you supply the extra proton to OH function, it can be converted to water molecule, and it is very easy to remove that from the molybdenum center, because always we know that the water center which is present to any metal center is very loosely bound. You can substitute that water by some other group. So, this is xanthine oxidase, then we can have the sulfite oxidase, which we can abbreviate as SO, and this basically has a molybdenum oxo center, then some sulfur center from cysteine sulfur, the other one is OH or water, and obviously the other two are double sulfur from the pterine unit. So, this pterine unit always basically fixing the molybdenum to the biological system, and that particular one is very much useful to go for the different eukaryotes and the prokaryotes, because in that particular molecule what we have seen that you have a terminal phosphate group. This terminal phosphate groups is very important, and sometimes there are other substitutions basically, because if we consider this O as OR. So, this phosphate terminal can be R. So, depending upon of these different substitutions, this R can be your H. So, in different eukaryotes this R will be H only. So, it is basically a free phosphoric acid type of attachment, but in case of the different bacteria and all, such as in prokaryotes, you have the different R groups. The R groups can be the different dinucleotides also. So, you can attach through these phosphate groups the different dinucleotides, that means the different bases and their corresponding monophosphate units, the cytosine monophosphate, the adenine monophosphate, the guanine monophosphate and all these, because one phosphate group is already available. So, you have the molybdenum center, then the corresponding pyranotherine unit, and that pyranotherine unit is useful for the attachment of the molybdenum, but your phosphate group can also play some important role to attach the different nucleotides or the simple phosphate unit. So, in these eukaryotes, your R group on that phosphate is basically H, and in case of the different prokaryotes, you have the nucleotides or such as dinucleotides. So, these groups basically can be attached to the molybdenum center. So, how these dinucleotides are attached? They are attached through phosphates. Through the phosphate groups. So, we are talking about 3 such families. So, one is xanthine oxidase, another is sulfide oxidase, and other one is Dmso Reductase. Which is abbreviated as DMR. So, in this particular case, so the corresponding coordination environments are basically different. So, here we are attaching all 6 positions. So, these are common to all others, that means their sulfur sulfur coordination from here, and on the 2 other sides also. So, it is like our acetyl acetonate molecule. So, on the both the 2 sides you have the sulfur sulfur binding from that terene unit, and you have Y and X. So, these groups are different. So, these what are these X and Y functions? These X and Y functions can be oxygen, sulfur, that we have seen in these 2 cases, oxygen and sulfur, here also oxygen and sulfur, and sometimes it can be selenium also, because we know that there are selenocysteine. So, from the selenocysteine moiety you can have the selenium group from over there, and this oxygen also it can vary a lot. Already we have seen it can be your oxido, it can be your hydroxido, or it can be your water. So, you can have O2 minus these oxygens, it can be HO minus, it can be water, and above these 3 you can have the free amino acids also, sometimes the serine residue, the carboxy function of the amino acid, and aspartic acid also. So, these are the 2 available. So, found also structurally characterized also, that they are the typical attached corresponding acid functions. That means, these 2 are basically going for oxygen for COO minus units. So, these COO minus units are coming from there. So, these 2 groups basically are completely different when we think of some selenium unit or the sulfur unit, but otherwise they are very much similar to that of your diaxomolybdenum acetyl acetone unit. So, you have 2 bidentate molecules, and these 2 bidentate groups are attaching to the molybdenum center, but the reactivity pattern of these 3 families of molecules are completely different, because attachment of these bidentate group, 2 such bidentate groups is basically stabilizing this particular center much more, because you have these 2 sites which are reactive, but compared to this particular one, these 2 centers are pentacoordinated. So, only one side you have the fixed coordination from the disulfur units, but you can have the 3 groups from the other 3 sides, and you can go for the change for the hydroxylation reactions or other reactions based on molybdenum. So, this particular biosynthetic part, little bit we should know that we have the molybdoterdin unit is begins with guanosine triphosphate. So, structurally how we will see that how you can have the guanosine triphosphate, and for this biosynthesis, because how you get this molybdoterdin, okay, this molybdenum center is responsible for giving some enzymatic functions, but initially the generation of this molybdoterdin is also interesting. So, two enzymatic reactions basically convert this triphosphate to cyclic phosphate of pyranoterdin. So, guanosine, basically your source for this nitrogen bearing part of the molecule is your guanosine. So, initially you have guanosine triphosphate, and that guanosine triphosphate is giving you a corresponding cyclic phosphate of pyranoterdin. So, if that cyclic phosphate part is lost, you will end up with the pyranoterdin part. So, this intermediate one, which is your pyranoterdin, is converted to molybdoterdin by the action of three further enzymes. If you just remember all these enzymes, the actions of these enzymes, little bit if we can know that, you see that all these enzymatic reactions, even the generation of this particular center is so interesting that here you require two enzymes for the conversion, that means the phosphate conversions, and after that how you get that particular molybdoterdin part, that means without molybdenum the ligand part. So, there also basically three further enzymes are utilized, and in dithiolate you should recall it that you have a double bond. So, you have a double bond part. So, this is a double bond. So, in dithiol part, so you have a double bond part and you have the dithiol. So, what we are generating on the system, you are generating a in dithiol part, because if this particular part is saturated one, you have much more flexible bidentate ligand, but when you have the double bond, it is much more rigid, and the planar structure is conserved when it is going to bind your molybdenum center. So, that is why it has some different role compared to ethane dithiol. So, you have all the ethane dithiol, the synthetic molecule is available in the laboratory. You can utilize ethane dithiol to go for molybdenum coordination, and you get the corresponding molybdenum compound. So, although the substituents on sulfur remain unknown, that is why I am trying to tell you that what are the R groups. The R groups is people are unable to identify, because during the processing this R groups is not available, it is only the molybdenum bound sulfur centers are available. So, what are the original R groups, whether they are the only hydrogen or some alkyl group, it is not known. So, sulfur is conveyed from cystenyl part sulfide. So, any cystenyl based molecule is there, and it is in situ if we can generate some part sulfide system, and this part sulfide system we all know in a double bond or triple bond it can go for the attachment to the corresponding organic backbone. So, this basically is very much similar to the synthesis of our iron sulfur proteins, because the sulfur is giving there and the cysteine sulfur is available for binding to your ferredoxin molecules. So, the monophosphate is adenylated in a step that activates the cofactor to a binding of molybdenum and tungsten. So, this particular ligand is now available to you and is basically activating the system for coordination, and when these metals are available, they are not in the free form, they are oxoanions, that means they are in the molybdate form and the tungstate form, and when they are in the molybdate form and tungstate form, they go and bind to the molybdoterdin cofactor. So, in situ we are generating this molybdoterdin cofactor, then little bit we can modify through those two sulfur, because the sulfurs are required. You do not able to utilize the oxygen and nitrogen for molybdenum coordination. That is why this, like that of your iron sulfur coordination chemistry or the cluster chemistry, the molybdenum sulfur chemistry is also very important. So, that molybdenum sulfur chemistry plays some important role, because whenever you attach sulfur atoms in the thiolate form, it can give you the corresponding reactions related to your molybdenum center. So, this basically what we are getting. So, this is what we are getting, that is guanosine triphosphate. So, what we have in your hand. So, basically if somebody asks you that how you convert the guanosine triphosphate to molybdoterdin. So, that is very simple enzymatic conversion. You should know how the molecule is getting transferred, because these are very recent knowledge for us, that how this particular part. So, this is there. So, you have the sugar unit and the triphosphate unit. So, this basically giving you a cyclic pyranopterin monophosphate, CPMP. So, these abbreviations are also nowadays very much important, because sometimes we find that some reactions we write that they are CPMP dependent reaction. So, it is nothing but cyclic pyranopterin monophosphate. So, you see that because it is basically the typical conversion for the monophosphate to diphosphate and triphosphate. So, ATP, AMP and ADP type of conversion. So, phosphate groups are getting hydrolyzed, and you have the sugar unit, and that sugar unit basically utilizing for this corresponding to giving you the pyranose unit. So, this pyranose ring is formed from this particular sugar unit, and this pyranose group, then you have these two positions available. You see that one is coming from the cyclic phosphate part. So, you have these two available groups or available points which are there and which are there for the corresponding conversion to N-diethyl unit. So, this enzymatic function, though it is very difficult to make in the laboratory, so the enzymatic reaction will push us to give you something where this particular part is getting modified through the insertion of two sulfur atoms, so that it can be a very good ligand with a part which is derived from guanosine molecule. So, this biosynthetic pathway will tell you how we can go, or basically we call it depending upon your demand, because we need a diethyl ligand, and that diethyl ligand is generating, because initially what people identified that initially we identified the molybdenum center first. The molybdenum is required for all these enzymatic reactions, and then we identified first. Then we tried to identify the immediate environment around molybdenum, what I have seen just that depending upon the different enzymes what are the molybdenum environment, and then people have identified the ligand part, and this ligand part is also very much important, but how it can be generated, that means people also able to trace that the different enzymatic steps, whether you require 2 enzymes in one step or 3 enzymes in the other step, and you require the corresponding groups as the cysteine persulfide, the requirement of the cysteine persulfide, how the sulfur is attached to the synthesis, because this sulfur attachment to the biological system is very much similar to that of your iron sulfur protein formation. So, this basically give you that ultimately we can say, and we can try to draw the structure, that this is the ultimate ligand structure which can go and bind to your molybdenum center. So, this basically reactions are all therefore is dependent on CPMP molecule, and that CPMP molecule is generated in situ from the particular molecule which is dependent on molybdenum. So, how these things are very much important, because once we identify that molybdenum is essential and its corresponding cofactor is also. So, deficiency also give rise to some problems to us. So, the deficiency of molybdenum cofactors, the early seizures, low blood levels of uric acid and high levels of sulfite, xanthite and uric acid in urine. So, if you can go some corresponding assimilation reactions where we get the corresponding nucleobases, the formation of uric acid. So, that we will see in case of our xanthine oxidase case, that the corresponding concentration of uric acid. So, uric acid concentration will vary from one particular part to the other. That means, your nutrition for all these nucleotides are not right. So, you have some problem. So, it can go for some diseases, and that diseases can be diagnosed by MRI images, and which can help you for the proper diagnosis. So, this is one such example. I am just citing out one example, because how people are doing, you see, is only very recent example, is only 2009 people have identified that is CPMP. So, it is well known for this family of and these doctors basically, that this particular CPMP concentration. So, if we have this in our body, so in Monash Children's Hospital people have identified in Australia that one baby is treated with molybdenum K-factor deficiency type A. We know, we consider all about your iron deficiency, all other things what is deficient to us. So, because these are very important. So, they have identified very nicely that they have molybdenum cofactor deficiencies, not molybdenum deficiency. So, not metal ion deficiency, not iron deficiency, but the corresponding heme deficiency. When we consider that, sometimes we call that you are deficient in hemoglobin, but whether you are deficient in iron or whether deficient in the globin part or the heme part, that is very important. So, this molybdenum cofactor, what is that? That molybdenum cofactor is without molybdenum the other part, the other organic part. So, they have identified that this that particular baby is deficient in molybdenum cofactor, and one particular type they have identified or labeled as a type A. And how you get the corresponding remedy? Okay, they are deficient in this particular one, so they are unable to produce the corresponding ligand which can bind your molybdenum center. So, you supply it externally. So, that is why they have been treated by this CPMP molecule. So, all these nucleo based type molecules, the sugar based, all these we can make in the laboratory. So, if the baby is deficient, so only important thing or the challenging part of this particular type of experiment is that you try to identify whether that particular patient or the baby is deficient in molybdenum or the corresponding cofactor. So, cofactor deficiency is very important to know. So, that particular thing is very costly thing that people can afford. In Australia also we cannot afford. They require daily injections of CPMP for the rest of our life. So, that is very important, but they can try, even if they can survive that baby for say 10 years or 15 years. So, this basically is a research finding, and they know it that it is not the molybdenum, but your ligand part. So, that is why they are also very much interested to know the biosynthetic part of the corresponding synthesis of the ligand, because we do not know, because we know the degradation of the heme part and the formation of the heme part, but we do not know much about all these particular processes when it is related to your molybdenum center. So, the one by one we will just see. So, the first one is your Dmso reductase. So, 3 families we have identified. Is that your Dmso reductase. So, in this Dmso reductase, how we get this corresponding formation of Dmso molecule through the supply of this oxygen from basically the water molecule. So, again it is a molybdenum containing enzyme capable of reducing your dimethyl sulfide or sulfoxide. So, if you can have the corresponding dimethyl sulfide, so you can reduce it from dimethyl sulfoxide to dimethyl sulfide, or sometime if you are able to get its corresponding dimethyl sulfone SO2. SO2 is the corresponding sulfone form. So, basically what we are getting, we are removing this oxygen, So, if it is molybdenum based, so if a molybdenum center is available which is sort of one oxo center, so this DMSO can convert that molybdenum center to its corresponding molybdenum dioxo form. So, that is basically its simple reaction. So, is a terminal reductase under anaerobic condition in some bacteria, because this anaerobic condition you do not require dioxygen, and that also immediately tells us that dioxygen from air or dioxygen from any other source is not required for this type of reaction. So, this is basically DMSO being the terminal electron acceptor. So, this DMSO molecule is accepting 2 electron and 2 proton. So, in any such reaction change, any biological reaction change, sometimes we require that there should be some useful groups or some useful mechanism where a substrate is available which can be functioning there as a very good electron acceptor. So, during the course of the reaction, the oxygen atom in DMSO is transferred to molybdenum. So, that is the basic idea, that how you transfer this oxygen from DMSO to the molybdenum center. So, that molybdenum centers definitely should not be in the corresponding form, that means it is not a corresponding oxo form or the diaxo form. So, what we have seen in case of its corresponding molybdenum active site, just now we have seen the corresponding molybdenum active site of Dmso reductase is that it has no oxo or the diaxo form. So, this particular form, so immediately it will have some bigger affinity for attacking this oxygen and assimilating as the molybdenum oxo or the diaxo species. So, this active site like that of our simple molybdoterine molecule containing the core supported by molybdenum in +6 oxidation state, and it can cycle between, already we have seen, in +4 and +6 oxidation states. So, the mechanism now we can immediately say that you have the involvement of the molybdenum in +6 oxidation state, you have the DMSO and the enzyme and the two possible oxidation state, and we will see that what type of coordination environment can stabilize the molybdenum center in +4 oxidation state and what type of environment can stabilize the molybdenum in +6 oxidation state. So, this is one such simple catalytic path, that when you have this particular one, that means the corresponding these two ligands, because this is the Dmso reductase has two ligands, in all two other cases you have only one particular diethyl group or diethyl ligand system attaching to the molybdenum. So, you have this molybdenum center which is in the +6 oxidation state and this oxo and this serine oxygen. So, these two are your X and Y. Just now what I have told you, we have written you that this particular DMSO center is based on something where you have two diethyl units like two acetyl acetone groups, and two of these groups are X and Y. So, one X is your oxo and another is your serine oxygen. So, this particular one, and that immediately when you are supplying electron and proton, basically this oxido function can be converted to hydroxido and then to water molecule. So, this basically is giving rise to that corresponding water molecule. When you transfer these two electrons to the molybdenum center, converting this molybdenum 6 to molybdenum 4. So, that is why this molybdenum center is settling between +4 and +6 oxidation states, and 2 electron step is therefore stabilized. So, immediately what we see that whether you are going from +6 to a +4 state or a +4 state to a +6 state, because this particular one, like that of your palladium in different catalytic system, when palladium in +4 oxidation state, we all know that it will be in a hexa coordinated environment. So, is basically oxidative addition, and in other case we get the reductive elimination. When we go for the reduction of the same molybdenum center, we go for reductive elimination. That is why you are moving from a hexa coordinated system to a penta coordinated system. So, this penta coordinated system is very useful therefore. So, when we basically reduce the system, that means from molybdenum +6 to we are going to a molybdenum +4, you are supplying electron, that means this you can do also electrochemically. Suppose you do cyclic voltammetry and you take this particular molecule, and the corresponding cyclic voltammogram will tell you that whatever species you are generating after reduction, because this you are going for a chemical reduction, but electrochemical reduction is also possible, and you are supplying 2 electron. And if you do the same electrochemistry in some protic medium, not in pure acetonitrile or pure dichloromethane, but in some medium where you have the protons, that means some aqueous medium also, because the removal of this oxo function you require this proton, that means the protonation is required for removal of this oxido function as hydroxido and ultimately as water. So, this hexa-coordinated species is going back to a penta-coordinated one. The cyclic voltammetric trace will immediately tell you that this particular environment, these two coordination environments are different, because this particular one is not going for only electron transfer, but some other group transfer, because this group is not there, this group is not present over there. So, this oxido function is not there. So, we also considered it as a some group transfer oxidation reduction redox reaction. So, once you reduce it, your oxo function is going and you are getting some penta coordinated one. That means, on the other hand, if you are able to generate some molybdenum species in low oxidation state, like that of your palladium center, platinum center or some other catalytic center in the zero oxidation state also, you can go for its corresponding reaction with dimethyl sulfoxide. So, dimethyl sulfoxide can nicely transfer this oxygen to your molybdenum center. This blue oxygen atom is immediately transferring to the molybdenum center, because what we get sometime that water molecule can give rise to a corresponding molybdenum oxo compound. Some hydrogen peroxides are also available for going for this, but if you utilize this particular one, because people have accidentally sometime discovered all these things, then they routinely followed all these things, because why we are bringing the question of measurement in the form of electrochemical measurement or cyclic voltammetry, because this particular species is a useful solvent. Because most of the time when we go for any studying molybdenum compound, the electrochemical measurements, if the corresponding compound is not soluble in dichloromethane, if it is not soluble in acetonitrile or even in DMF, we try dimethyl sulfoxide. So, dimethyl sulfoxide is a routine solvent for all these sort of measurements, but remember, we should remember very nicely that this particular one that is not reacting with the compound of this type. If it is reacting, you will end up with some dimethyl sulfide, and you are able to identify the corresponding dimethyl sulfide from the medium. It has some typical smell also, and from the solvent medium you can recover dimethyl sulfide, which can also establish that during catalytic cycle involving electrochemistry also, cyclic voltammetry also, you can generate some amount of dimethyl sulfide from your solvent, which is your dimethyl sulfoxide. So, that is why related to that is very important. So, next day, because today we are not able to cover this one also. So, the second one, that sulfite oxidase we will study. So, in this particular case is enzyme. So, definitely again like that of our dimethyl sulfoxide thing, that means you can transfer this oxygen. So, sulfite to sulfate. And what we all know, because these are very useful biological system and they are very mild system, and sometimes we get to know that they are going via some cytochrome C, which basically your source of electrons, because we are not using some any other chemical reducing agent or the electrode system for this electron transfer, and this transfers the electrons produced to the electron transport chain. So, we all know that cytochrome C oxidase, all they are present in cytochrome C electron transfer chain. So, if at some point we require so many electrons, that means that we require 2 electrons to activate the molybdenum center, and that activation for the molybdenum center is useful for the oxidation of sulfite to sulfate unit. So, at the same time, like since we all know that the corresponding electrode potential, that how much free energy you are transferring from there. So, basically we can also allow the generation of the corresponding ATP. So, phosphate transfer potential is useful. So, phosphate transfer potential, if it is available for this electron transfer reaction of conversion of sulfite to sulfate, at the same time as a byproduct definitely. So, as a byproduct we should be able to produce some amount of ATP molecules. So, basically we are very much getting some useful molecules like ATP during the corresponding oxidation of sulfite to sulfide. So, it is again a molybdenum dependent enzyme, and the same molybdoterine cofactor is utilized for the conversion of this reaction, and now which is not there in case of dimethyl sulfoxide type of operation or using molybdenum enzyme, that heme group is required. Where is heme group? That is also a system, also a reaction where it is dependent on cytochrome C. Since it is cytochrome C dependent, therefore, the corresponding sulfite oxidase should also be dependent on your heme group. That means, this is dependent on two such metalloenzymes. One metalloenzyme is involving molybdenum and other part is involving your iron center. That means, iron heme which is present in cytochrome C molecule is also involved for sulfite oxidase. So, you can have the detailed information from this reference for this molecule. So, little bit I will just carry on next day also, that this is the whole complicated molecule, and this person is well known for this molybdenum sulfur chemistry, John A. Enemark. So, even if you go for his site, I have taken it from his site, basically his home page. So, you will get this particular information, that they have the corresponding protein structure for the entire molecule, and when they crystallize this particular thing, because what we see that during the determination of this all entire protein structures. So, protein structures and the protein structure solutions are sometimes very difficult without metal center. So, we should be very happy if you have a metal center over there. That means, solving the structure of only the corresponding ligand part without metal center is sometimes difficult, because the scattering factor related to the heavy metal or the heavy center, one particular technique is well known, that the heavy atom method. So, we are lucky enough if you have a corresponding metal center within the protein structure, it is very easy to crystallize it. It is helping your crystallization process, because until and unless you do not get the good crystal, you do not have the corresponding protein structure. And in this particular case, what we will see, that during this conversion basically is not that particular metal center is available, the metal centers are there definitely depending on the molybdenum and iron, but this particular anion, when it is forming from the sulfite function and it is converting to sulfate, and that sulfate is basically trapped inside. So, sometimes we will just do not worry for that, we will come back again in this particular one. So, you see that the product, that means sulfite has been converted to sulfate and that is being trapped within the system. So, this particular system is very easy, or you can characterize it, I can crystallize it and identify the molybdenum center as well as the sulfate center very nicely. So, this basically system, because we are studying so much about the corresponding anion recognition and all these things. So, now anion which is produced from the system through the oxidation of sulfite is now being trapped within the molecule itself. So, molybdenum center is there and sulfate what is formed is getting trapped. Okay. Thank you.